Okay, so welcome back to this next video in which we are discussing the PI3 kinase AKT mTOR pathway and its involvement in cancer. Okay, right, so we've discussed that type 1 PI3 kinases are heterodimers which consist of a catalytic subunit and a regulatory subunit. Okay, and the catalytic subunit, there are 4 different possibilities which are called the P110 proteins. Okay, so there's P110 alpha, P110 beta, P110 gamma, and P110 delta. Okay, and these are coded for by genes which are called PIK3C for catalytic subunit, and then you have the corresponding Roman alphabet letter for the Greek alphabet letter here. So A for alpha, B for beta, G for gamma, and D for delta. Okay, right. We've then discussed that the type 1 PI3 kinase is named after what catalytic subunit it's made out of. So there are PI3 kinase alpha, beta, gamma, delta, corresponding to the 4 different catalytic subunits that you can use to build your type 1 PI3 kinase. Okay, and we've then discussed that the type 1 PI3 kinase enzymes can then be further divided into type 1a PI3 kinase enzymes, which are the PI3 kinase alpha, beta, and delta, and these ones are all going to have certain regulatory subunits that can bind to them, and this regulatory subunit is also going to be able to bind to receptor tyrosine kinases, and therefore these PI3 kinases, the type 1 PI3 kinases, are going to be activated by receptor tyrosine kinases. Meanwhile, the type 1b PI3 kinases, including only PI3 kinase gamma, that has very different regulatory subunits which can bind to it, as we'll discuss, and those regulatory subunits aren't capable of binding to the receptor tyrosine kinase, and therefore type 1b PI3 kinases aren't activated by receptor tyrosine kinases. So let's now turn our attention onto the regulatory subunits of PI3 kinases then. Okay, so let's start off with the regulatory subunits that can be used in type 1a PI3 kinases. So remember, if we're making a type 1a PI3 kinase, that means that we've either used P110 alpha, P110 beta, or P110 delta to create PI3 kinase alpha, beta, or delta. Okay, right. So, the regulatory subunits that can be attached to these catalytic subunits are called P85 type regulatory subunits. Okay, and there are 3 different genes which can be used to make these P85 type regulatory subunits, and one of these genes has 3 different splice variants. So let me firstly tell you the names of the genes which make the P85 type regulatory subunits, and then I'll tell you the names of the proteins which they can produce. Okay, right. So the genes for P85 type regulatory subunits are called PIK again, and then you've got the 3. And now R for regulatory subunit, so PIK3 is for PI3 kinase, R for regulatory subunit, okay, and then you've got R1, then you've got PIK3R2 and PIK3R3. So there are these 3 genes which code for P85 type regulatory subunits, which are the type of regulatory subunit that can be attached to P110 alpha, P110 beta, or P110 delta, which are the catalytic subunits in these type 1a PI3 kinases. Okay, right. So PIK3R1 has 3 different splice variants that it can produce, and these 3 different splice variants have very different molecular weights. Okay, so the 3 different splice variants it can produce are of 85 kilodaltons, and this is called P85, and then you put an alpha there to show that it's from this type 1 regulatory subunit gene. Okay, it can also produce one of 55 kilodaltons called P55 alpha. Okay, again, the alpha denotes that it's from this R1 gene. Okay, and then finally it can produce one of 50 kilodaltons, which is called P50 alpha. So those are the 3 different regulatory subunits that are all P85 type regulatory subunits and can all be dimerized with these 3 catalytic subunits. And they're all created by this one gene, PIK3R1. They're the 3 different splice variants of PIK3R1. Okay, right. Then for the gene PIK3R2, this only has one splice variant, okay, and again it weighs 85 kilodaltons, so this is P85, okay, and because it's coded for by this second gene, we put a beta there, okay, so P85 beta is an 85 kilodalton regulatory subunit of a PI3 kinase made by this PIK3R2 gene, okay, and it's the only one that's made by that gene. Finally, the PIK3R3 gene, that makes a regulatory subunit of 55 kilodalton molecular weight called P55 gamma. Okay, so these 5 different proteins here, P85 alpha, P55 alpha, P50 alpha, P85 beta, and P55 gamma, these are the P85 type regulatory subunits which can be bound to one of these catalytic subunits here to make a type 1a PI3 kinase. Okay, so to summarize here, when we're building one of these PI3 kinase heterodimers and we want to build type 1a PI3 kinase, you put a P110 subunit here, and that will either be P110 alpha, beta, or delta, and then you put a P85 type regulatory subunit here, and that will be one of these 5 proteins here, either P85 alpha, P85 beta, P55 gamma, P55 alpha, or P50 alpha. Okay, so one of those 5. Again, that's often how people will show it in little cartoons. They'll just show P85 in a box to denote a P85 type regulatory subunit, and then P110 in a box to show a P110 type catalytic subunit. Okay, right. Now let's talk about the class 1b, or the type 1b PI3 kinases. Okay, so remember the type 1b PI3 kinases, they had as their catalytic subunit always P110 gamma. Okay, now they have different regulatory subunits. Okay, so P110 gamma does not bind to the P85 type regulatory subunits. Okay, it has only 2 regulatory subunits that it can bind to, and these are called P101. Okay, and also P87. So it has 2 options for what it can bind to. It can either bind to P101 or it can bind to P87, and these are coded for by different genes. So P101 is encoded by PIK3R5, okay, and P87 is encoded for by PIK3R6. I don't quite know what happened to PIK3R4, okay, but these are coded for by PIK3R5 and PIK3R6. Okay, right. So, if you want to build a type 1b PI3 kinase, you don't have any choice about the catalytic subunit. Okay, so if we draw this heterodimer here, you don't have any choice about the catalytic subunit. That is forced to be P110 gamma, but you do have a choice in the regulatory subunit. It can either be P101 or it can be P87 there. Okay, and that's a type 1b PI3 kinase enzyme. Okay, right, now before we look at the structure of these regulatory subunits and catalytic subunits in a bit more detail, and don't worry, we're not going into it in too much detail, we're only going into the detail that we're going to need for some of the later comments I want to be able to make, okay? And before we go on to that, let me just say something about the expression profiles of these different types of type 1 PI3 kinase enzymes. Okay, so basically, you find PI3 kinase alpha and PI3 kinase beta type 1 PI3 kinases all over the body. Okay, so these are expressed pretty much everywhere. In all cells of the body, you're going to find PI3 kinase enzymes where the catalytic subunit is P110 alpha or P110 beta, and the regulatory subunit is therefore a P85 type regulatory subunit. Whereas, in contrast, the PI3 kinase gamma and PI3 kinase delta enzymes are much more selective in where they are expressed. Okay, they are generally found in white blood cells. Okay, and we're going to find that this is very significant later on for leukemia. Okay, so the major drug that has had success so far as in blocking this PI3 kinase AKT mTOR pathway is used to treat chronic lymphocytic leukemia, and the way it actually works is by inhibiting PI3 kinase delta. So we'll talk more about that drug later on. It's called idelalisib. Okay, but we'll come to that later on. That's the major success story that's spurring on more research into drugs which inhibit PI3 kinase AKT mTOR pathways. Okay, right. So PI3 kinase alpha and beta, they're found everywhere. PI3 kinase gamma and delta, they're much more selective in where they're expressed, just in leukocytes, white blood cells. Leukocytes is just the fancy term for white blood cells, WBCs. Okay, right. So let's now look at the structure then of type 1 PI3 kinase subunits in a little bit more detail. So the first thing that I want to talk about is the structure of one of these type 1A catalytic subunits in more detail. So I want to look at P110 alpha, P110 beta, and P110 delta. Okay, and we're just going to look at its primary structure. So we're going to see some of the key domains that P110 alpha, beta, and delta have, and we're going to see the importance of certain of these domains later on in the video. Okay, so for now, here is the amino terminus of the P110 alpha, beta, or delta protein. Okay, and the first important domain that you have in the protein is what's known as a P85 binding domain, and this is the domain which is used to bind to the P85 type regulatory subunit. Okay, so this for short is just abbreviated to P85BD for P85 binding domain. Okay, so I'll show you the portion on the P85 type regulatory subunit later that actually binds to that P85 binding domain. Okay, then after the P85 binding domain, what you have is a domain that's known as the RAS binding domain. Okay, an RBD, and we're going to see the importance of this later on, okay, when we want to discuss the activation of PI3 kinase enzymes by small GTPases. One of the key small GTPases that is capable of activating many PI3 kinase enzymes is RAS, okay, so we'll come back to this as well. So this is a RAS binding domain, so I'll just write that one out in full. So the R is for RAS. Okay, then the BD, I'll just abbreviate down to BD, binding domain. Okay, right. Then after the RAS binding domain, you have a domain that's found in many different proteins called a C2 domain. Okay, now C2 domains are generally found in proteins that bind to calcium and are activated by calcium. So for instance, it's very important in protein kinase C enzymes. Okay, what its importance is in the catalytic subunits of P110, catalytic subunits of PI3 kinase enzymes isn't fully known yet. Okay, so there's a C2 domain. Then what you have is a helical region made up of alpha helices. Okay, so this is a helical portion here. And then following the helical portion, you then have the actual catalytic domain of the catalytic subunit. So just to reiterate again, this is the primary structure of either P110 alpha, P110 beta, P110 delta, i.e. the catalytic subunits of type 1A PI3 kinase enzymes. Okay, so there's the helical domain, and then here in vivid purple, that's the catalytic domain of this catalytic subunit. So this is the domain that actually catalyzes the phosphorylation of PIP2 into PIP3. Okay, so that's the domain primary structure of a type 1A catalytic subunit of PI3 kinase. Okay, now, obviously it would be folded up in reality, it wouldn't be linearly like this. Okay, but that shows us some of the important domains that we're going to need to understand when we want to discuss the activation of these later on. Okay, right, so that's the structure, the domain structure of P110 alpha, P110 beta, and P110 delta. The next thing I want to make a comment on is how is P110 gamma then different to these. Okay, well, it's not hugely different. All that differs between P110 alpha, beta, and delta and P110 gamma is that you have lost this P85 binding domain. Okay, so that's important because that now means that the P110 gamma cannot bind to the P85 type regulatory subunits. So basically, if you wanted to draw P110 gamma, just get rid of this bit, scrap this. Okay, start the amino terminus there and just have these portions here. That would be P110 gamma here. Okay, right. Next thing I want to comment on is the structure of a P85 type regulatory subunit, because this is actually known. Okay, we have crystal structures of what the three-dimensional structure of this looks like. Okay, now we're not going to look at the three-dimensional structure of it, we're just going to look at its domain primary structure. Okay, but that's an achievement because we don't have that for the P101 or the P87 yet. Okay, so I'm not going to be able to show you what the domain structure of these two is, but I can show you what the domain structure of these is. Now this is where it differs depending on what size you are. Okay, so the P85 ones are longer than the P55 ones and the P50 ones. Okay, and these have additional domains that these three don't. So P85 alpha and P85 beta are bigger, they have more domains than P55 and P50. Okay, the P55s and the P50, they have all the same important domains. Obviously P50 is going to be smaller, but it doesn't lack any of the important domains that P55 alpha and P55 gamma have. Okay, so we can summarize those two together. So I think I'll go down here to show you the structure of a P85 type regulatory subunit. Okay, so this first portion that I'm about to put here, this obviously isn't essential for the function because this is the portion that is only going to be there in the P85 alpha and the P85 beta, but not the smaller ones. Okay, right. So this first bit consists of an SH3 domain, okay, another very famous domain found in a huge number of different proteins. Okay, it stands for Sark homology free domain because it was first found in the non-receptor tyrosine kinase Sark. Okay, so SH3. Then after the SH3, what you have is a proline-rich domain, which I'll just abbreviate down to P here. So that's a proline-rich domain there. Okay, and I'll just put a few labels here. So SH3 stands for Sark homology. Okay, so the S is for Sark, H is for homology, and it's the Sark homology free domain. And we're going to see another example of a Sark homology domain in a moment that's going to be more important for the function of the P85 subunit. Okay, this P domain here, this is a proline-rich domain, so it has a lot of proline residues within it. Okay, right, then the final domain that's in this little extension out the front that you have in P85 alpha and P85 beta, but not in the other three, is what's known as a breakpoint cluster homology domain. Okay, BH domain, which I'll colour in blue here. Okay, so BH stands for breakpoint cluster homology domain, and again it's a domain that you find in many different proteins, and the function of it here isn't fully understood yet. Okay, so breakpoint cluster homology domain. Right, so those are the three domains that you only have in P85 alpha and P85 beta, but not in the final three. So those can't be essential for their function as a regulatory subunit of PI3 kinase, catalytic subunit. Okay, right, so now we start the essential bits, and these are present in all 5 of them. Okay, so if we were talking about one of the smaller ones, such as P55 alpha, P55 gamma, or P50 alpha. Okay, so P55 alpha, P55 gamma, and P50 alpha, they would start now. They'd have their amino terminus here, and they'd have all of the bits that I'm about to tell you about. Okay? So, again, I'll stress the point that although P50 alpha is, of course, going to be smaller than P55 alpha, it doesn't lack any of the important domains that P55 alpha has, okay? And that's why, as far as my very simple domain structure picture is concerned, they're going to look the same, basically. Okay, right. So now what we have is another proline-rich domain. So this breakpoint cluster homology domain is flanked by 2 proline-rich domains in the P85 alpha and the P85 beta. In these smaller ones, the proline-rich domain is the first domain that you have in this P85 type regulatory subunit. Okay, then what you have is two SH2 domains split by a domain that sits in between them called the inter-SH2 domain. Okay, so this one we'll call the NSH2 domain. All that means is the N terminal, the one that's closer to the N terminal, and then SH2, again, it's a really famous type of domain, Sark homology 2 domain. Okay, again, a domain that's found in a huge number of different proteins, named after the fact that it was first found in the Sark non-receptor tyrosine kinase, but has since been found in loads of other proteins. Okay, so this is a Sark homology 2 domain, and this is what we're going to call the NSH2 domain, simply because there are going to be two of them, and this is the one that's closer to the N terminus. Okay, now Sark homology 2 domains are really important in binding to phosphorylated tyrosine residues on, for instance, receptor tyrosine kinases that have been activated, and indeed this is going to be what the important function of this is, that it's going to play an important role in the activation of the PI3 kinase enzyme by receptor tyrosine kinases. Okay, then what you have is a domain called the inter-SH2 domain. So this is not an SH2 domain. This is standing for the inter-SH2 domain. So ISH2 here stands for inter-SH2 domain. Okay, and it actually consists of two alpha helices that end up folding back on one another and coiling into a coiled coil domain. Okay, right, so that's the domain sitting in between the NSH2 domain and the final important domain of this P85 type regulatory subunit, which is the C-terminal SH2 domain, the CSH2 domain here. Okay, again, this is another Sark homology 2 domain, and because it's the one that's nearer the C-terminal, it's called the C-terminal SH2 domain. Okay, right, so these are the two really important domains here in these P85 type regulatory subunits. These are the ones that are going to bind to phosphorylated tyrosine residues. Okay, so again, I'll reiterate the point that if you are one of these smaller types of P85 type regulatory subunits, either P55 alpha, P55 gamma, or P50 alpha, then you're just going to start here and you're just going to have this proline-rich domain. Then you've got these two SH2 domains, an N-terminal and then the C-terminal SH2 domain with the inter-SH2 domain in between them. Okay, now the next important thing I should tell you is how does the P85 type regulatory subunit bind to the type 1a catalytic subunit? Okay, well, it's through an interaction between this inter-SH2 domain here and the P85 binding domain here. Okay, and remember, again, we've drawn them as linear primary structures. In reality, they'd be a folded structure. Okay, and this does resemble a blob when it folds up. Okay, so it's certainly not a great big long protein like we've drawn it. It folds up into a blob. Okay, right, so in fact, these pictures are a little bit more accurate, really, in what they actually look like. Okay, but it's through the interaction with the inter-SH2 domain that the catalytic subunit P110 alpha, P110 beta, or P110 delta binds to the P85 type regulatory subunit. Okay, right. So those are the structures of PI3 kinase subunits in more detail. Okay, and as I say, we've done that so that when we want to come on to discussing things like how they are now activated and mutations later on, where mutations are, we have these domains and we can point at where they actually are, so that we're not just saying, oh, and this is in the helical domain. We actually know where that is in the protein. Okay, right. So we'll call it there for this video, and in the next video what we'll turn our attention onto is the mechanism by which PI3 kinase enzymes, type 1 PI3 kinase enzymes, are actually going to be activated. So we'll see that they can be activated by G protein coupled receptors, they can be activated by receptor tyrosine kinases, they can be activated by small GTPases. And then after we've discussed their activation, we'll then see what the generation of phosphatidylanosyl 3,4,5 trisphosphate in the cell membrane is going to do, and that will lead us on to the rest of the PI3 kinase AKT mTOR pathway.